Good day, everyone. We are Group 3, comprising of Colmo, Diaz, Lauro, Suelto, and Tomaning. And today, we will be discussing the importance of the gut microbiota. So here is the outline of our group's report. First, we will be tackling the introduction of the gut microbiota. Second, are the examples of good microbiota. Next, are the factors affecting gut microbiota. And lastly, the effects of gut microbiota on human health. Before we do an in-depth discussion of the gut microbiota, it is important that we know its basic concept in order to identify its relevance to living organisms, specifically to us humans. A study from Turdsby and Job defined gut microbiota as bacteria, archaeas, and eukaryotes inhabiting the human gastrointestinal tract, and around 100 trillion microorganisms reside in the humans. The gut microbiota is composed by environmental factors such as diet and host genetics, wherein most yield similar protein or metabolite profiles. This gut microbiota and the human have co-evolved to form a symbiotic relationship, meaning that gut microbiota and humans mutually benefit from each other. Some benefits that microbiota offer to humans are maintaining the integrity of the mucosal barrier, protecting against the pathogens, regulating host immunity, strengthening gut integrity, shaping intestinal epithelium, and harvesting energy. Additionally, gut microbiota help humans absorb nutrients by breaking down complex molecules such as the plant cellulose.
So, examples of human gut microbiota. The first one is the Lactobacillus species. It's a lactic acid bacteria, a family of gram-positive facultative anaerobic rod-shaped bacteria. And the second one is the Bifidobacteria species, or the Bifidobacterium, a family of gram-positive anaerobic branch rod-shaped bacteria. So the Lactobacillus strain is the most important and predominant good bacteria that live mainly in the human small intestine, while the Bifidobacterium strain is located mainly in the walls of the colon to help prevent the spread of harmful bacteria and other pathogenic microorganisms. Strains of lactic acid bacteria are the most common microbes engaged as probiotics, especially Lactobacillus and Bifidobacterium species. Of the more than 100 Lactobacillus species, the most commonly used probiotics are Lactobacillus acidophilus, Fermentum, Plantarum, Rhamnosus, Paracasei, Gasseri, Reuteri, Salivarius, and Lactobacillus helveticus. And among the 30 bifidobacterial species, those recognized as probiotics include Bifidobacterium bifidum, longum, infantis, lactis, brev, and Bifidobacterium adolescentis.
Given that the human gut microbiome is a complex community of trillions of microbes that are constantly interacting with each other and our bodies, there are several ways, both intrinsically and extrinsically, for the microbiome to be changed or altered, which begins when we are infants. One of the first factors that determines one's gut microbiota network is DNA. According to a Cornell-led study, humans at birth generally have no microbes in their bodies. However, through breast milk, food, and their environment, gut microbiota networks quickly form, and the type of microbes that colonize a person's gut and how abundantly can be strongly influenced by human DNA. Some genes, in particular, impact the levels of microbes that thrive inside the gut and even influence a person's food preference and taste perception, which leads us to the second factor that affects the gut microbiome, diet. It also plays a role in influencing the kinds of microbiota that live in the gut, as an individual's dietary patterns and habitual intake play a role in shaping one's microbiota profile. For example, a high fiber diet affects the type and amount of microbiota in the intestines because dietary fiber can only be broken down and fermented by enzymes from microbiota. Medications such as probiotics and antibiotics also reshape and alter gut microorganism communities. Probiotics, which are defined by the Food and Agriculture Organization as live microorganisms, which, when administered in adequate amounts, confer a health benefit on the host, may restore the composition of the gut microbiome and introduce beneficial functions to gut microbial communities. After consumption, they have the capacity to prevent gut inflammation and aid the treatment of infectious and antibiotic-associated diarrhea and insulin resistance and diabetes, amongst others. Antibiotics, on the other hand, result in a variety of negative effects on the gut microbiota, including a reduced diversity of species, alteration in metabolic activity, and the selection of antibiotic-resistant organisms. Because they also destroy the good bacteria, an upset stomach and or diarrhea usually result after the consumption of an antibiotic.
We will now tackle the question, is gut microbiome the same for all people? So, a person's microbiota network is completely unique and is first determined by their DNA. Microorganisms are initially seen by a person when they are an infant, in the birth canal after delivery, and in the mother's breast milk. The specific species of germs to which the newborn is exposed exclusively depends on the species present in the mother. Later, dietary changes and environmental exposures can alter a person's microbiome, either for the better or worse in terms of health and disease risk. In conclusion, each person's microbiome is unique, and individual variations are significant in comparison to the normal long-term biochemical changes that occur within a person over time.
Now that we know what gut microbiota is, its examples, factors, and difference from one individual to another, let us talk about its importance in one's health. As mentioned earlier, a person's microbiota depends on their diet, which makes it unique to every individual. The immune system and gut microbiota have a mutualistic relationship, as the microbiota serves as an educator and stimulator of immune responses, while the immune system preserves this symbiotic relationship that they have. The microbiome also functions in breaking down compounds in the digestive system and synthesizing amino acids, especially B vitamins and vitamin K. Threading all this information together shows how significant microbiota is to the overall health of a person. Forming a healthy relationship with your microbiomes, for example, moderate intake of foods with probiotics avoids further digestive and autoimmune problems. That is why we must not be afraid of it, but instead thank our microbiomes for keeping us healthy and safe from pathogens entering our body through contaminated foods. And that is the end of our presentation. Once again, I am Diaz together with Como, Laro, Suelto, and Tomaneng, reminding you to keep your microbiomes healthy. We will meet again.